Good afternoon. I would like to welcome you back to the HIAAD seminar series called Bridge, because the idea of these seminars is to bridge different people from different expertise that they all focus on dementia and research on Alzheimer, both clinical practice and research in that field. And starting this new academic semester, we have a very great speaker. Cristina Dalla, Professor Cristina Dalla will introduce her. And thanks again for coming with us in this new year of HIAAD seminars. Cristina.
Yes. Hello. I wish you a happy new academic year. Okay. We wait for the switch.
Yeah, we always wait for the switch, but now we will have it. You see, everybody is on summer mood. That's why I put this t-shirt today. Trying to be still on summer mood. Unfortunately, it doesn't work because here we have some proposal to submit tomorrow. All of us are extra busy these days. But I think we are very happy to have Ivan with us. And are we ready to connect Professor Cristina Dalla? Not yet. So I should say something.
Thank you very much for those nice talks, the invitation, and also for the for the opportunity to present my research to your community and in this seminar series. So I will start sharing my screen. Let's see here. Yes, so I'm active at the Karolinska Institute in Sweden. As you heard, at the Department of Neurobiology, Care Science and Society, the Division of Neurogeriatrics. And I will talk about today our research about sex hormones and sex differences in Alzheimer's disease. And we'll also talk about data that we have already published, but a lot of this will be also unpublished data that I think is quite exciting. So first I want to just mention that, as you all know, that sex differences are found in disease risk. And maybe the most obvious are cancers in reproductive tissues that affect men and women differently, but also other diseases such as cardiovascular disease. These are more prominent in or prevalent in males at reproductive age, while osteoporosis, for example, is more prevalent in females at reproductive age. And the reason why we have these differences between disease risk can, among other factors, be ascribed to sex hormones. That in females, there's female sex hormone estrogen and progesterone, they drop at menopause. And this actually has been very well established that this correlates to increased risk of cardiovascular disease and osteoporosis. But are there also differences in neurological disorders? And well, yes, there are sex differences in neurological disorders. And this is a sort of a scheme that I often want to highlight, where we can see that males are more prevalent, or neurological diseases in males are ADHD, Tourette syndrome, autism. These are more prevalent in males, but females are more affected by Alzheimer's disease, depression, and anxiety. And for example, you can also see that these diseases are more neurological, neurodevelopmental diseases, while these are more age-related diseases. So what is the cause to these sex differences? Well, of course, the environment plays a huge role. Education, social interaction, cognitive stimulation, what you're exposed to, and so on. These can all modulate the risk for different sexes to get these neurological diseases. But also, this is called the gender dimension. But also biological sex is very important. So chromosomal differences and sex hormones in this case. These can modulate developmental neural organization, the immune response, and metabolism. And together, these both dimensions modulate the risk of these different neurological diseases. And what I study in my lab is sex hormones in mental health, but also in dementia. And I will talk about today is sex hormones and in Alzheimer's disease, which is a topic that we're very interested in and started to focus more and more during the last 3-4 years. And I don't need to really to explain what Alzheimer's disease is to this audience, but I'll just briefly go through this. As you know, this is the most common type of dementia. This is an age-related disease and progressive disease where 1 in 10 of all people over age 65 have Alzheimer's disease, and every third person over age 85 have Alzheimer's disease. Still, the causes are not really known, and 95% of all AD cases are sporadic, but there is an inherited component there. But which exactly genes, how they interact, is not really well known yet in the sporadic disease. But environment and family history of other diseases appear to play a big role. And we also know that carriers of a specific gene polymerase in the ApoE gene, these persons have a 2- to 3-fold increased risk of AD. So it's a complex interaction between genes, environment, and disease history. So AD is characterized by memory decline, misfolded amyloid beta depositions around synapses, called amyloid plaques, as you know, intraneural misfolded tau tangles, and also extensive neuroinflammation, which leads to neuronal cell death. There's no real cure yet, although there are promises maybe soon, but right now, early detection to implement preventive measures is very, very important to find. So what about sex differences in Alzheimer's disease? Well, on every male person that is diagnosed with Alzheimer's disease, there is 2 females that are diagnosed with Alzheimer's disease. So 2/3 of all AD cases are women, which can be illustrated with this very old, quite old study from Framingham. But this could be, since we know that Alzheimer's disease is an age-related disease, and we know that women live on average like 4 to 6 years longer than men, it could be just simply that women live longer and they have higher risk of Alzheimer's disease. It could be like that, and this probably is a bit like that. I would also know that women have better verbal memory, so they can mask the disease. So they are less women are actually being diagnosed with Alzheimer's disease. And when they are diagnosed later than men. But once they are diagnosed, women have a faster rate of cognitive decline. But it's not really only age-related. And maybe the most striking example of this is that women that are heterozygous for this ApoE, a specific ApoE variant E4, have a 4 to 10-fold higher increased risk of being diagnosed with Alzheimer's disease compared to men that have exactly the same allele. And estrogen has been reported to be neuroprotective, and we know estrogen is the female sex hormone. And loss of this estrogen means that you lose some resilience. And this happens at menopause, which can increase the risk of Alzheimer's disease then. And also some observational studies have suggested that menopausal hormonal therapy can reduce AD risk. So biological factors are involved, very importantly, and sex hormones, we think, can modulate AD risk and AD pathology. So to refresh your memory about sex hormones, there are 3 types of sex hormones. We have progesterone and estrogen, which are classical female sex hormones, and we have testosterone, also androgen, which is the classical male sex hormone. And they bind to nuclear receptor transcription factors that upon binding of the sex hormone, they bind to the DNA and mediate transcription of different genes. So in case of progesterone, it binds to the progesterone receptor. Estrogen binds to estrogen receptor alpha, estrogen receptor beta, or the G protein coupled receptor, estrogen receptor, which is not really a nuclear receptor, but a cell membrane receptor. And testosterone binds to the androgen receptor. And in case of progesterone and estrogen receptors, they participate in a lot of different gene regulations involved in development, growth, and of course, female reproduction, while the androgen receptor mostly regulates male reproduction. And something also that's very important to understand is that testosterone can be converted to estrogen in different tissues. And this happens very readily in the human brain. As I told you, there have been a lot of studies that have shown that estrogen is neuroprotective, that menopausal loss of estrogen may increase risk of Alzheimer's disease, and also that hormone replacement therapy may decrease risk of Alzheimer's disease. So the question here is, why don't everyone get estrogen supplementation, like all the women get estrogen supplementation during menopausal transition? And well, there are 2 reasons for this. One is that it's not really well characterized, or very well described, how hormone replacement therapy can decrease the risk of AD, or if it even does decrease the risk of AD, because there's not enough studies yet on this. Another is the risks with menopausal hormone replacement therapy. As we know, menopausal hormone therapy, it acts on the central nervous system and the reproductive organs to alleviate the stress and discomfort associated with menopausal transition, and also prevents osteoporosis. And there are 2 main components of menopausal hormonal therapy. This is progesterone and estrogen. And you take progesterone in order to avoid endometrial proliferation when you take the estrogen. So you have to, if you have an endometrium, like a uterus, you have to take also estrogen if you take hormonal therapy. But there are risks here. Progesterone is associated with increased risk of breast cancer. And estrogen, when it can bind to estrogen receptor alpha, which is very widely expressed, so you get many unwanted systemic effects. You also have increased risk of endometrial cancer and also risk of thrombosis. But estrogen also binds to estrogen receptor beta. And this estrogen receptor is only expressed in the central nervous system, in B cells of the B cells, immune cells, in colon and gonads, and it has to date no known adverse effects. And it's been shown to have actually anxiolytic effects, meaning that it can alleviate depression. And it's actually an ongoing trial, clinical trial now about seeing if targeting ER beta can alleviate menopausal depression. And there are nowadays a new generation of ER beta agonists called benzopyrans. These bind to ER beta with 13-fold selectivity over ER alpha and with 32-fold higher potency. So this is maybe not very selective actually, but what is important here is that when bind to estrogen receptor alpha, it binds the opposite orientation and inhibits the adverse effect of ER alpha in these reproductive tissues. So this is maybe the most clinically relevant ER beta agonist, that it actually counteracts ER alpha function in adverse ER alpha functions in other tissues. So could targeting ER beta actually be neuroprotective? Well, it's been shown by my old mentor, Jan-Åke Gustafsson, already in year 2000, more than 4 years ago, that ER beta knockout mice display fewer neurons in the cortex, but also smaller neurons in substantia nigra. And they say that there is a progressive neuron cell body degeneration in this aged ER beta knockout mice. So we decided to test this in an AD model if ER beta can alleviate AD pathology in an AD model. And we use this APP and LGF mice. These are knock-in mice we have knock-in of 3 mutations in the endogenous APP gene. We have a Swedish mutation, Iberian mutation, and Arctic mutation, which leads to quite rapid pathology from 2 months until 9 months of age. So at 9 months of age, they have very saturated pathology. And at 6 months, they have like intermediate pathology. So we used both males and females here, and we treated these mice with this very selective ER beta ligand, LY5000307 from Eli Lilly. We gave them the mice oral gavage over 5 weeks, and at 6 months we checked memory function, amyloid pathology, and neuroinflammation. And some females were also overectomized, meaning that we performed surgical menopause. So this is the work from one of my talented PhD students, Aphrodite Demetriou from Cyprus. She checked memory function in the mice. Here is spatial memory, and when we treat them with LY ligand, the ER beta activation, we see that both males and females perform better in the Y-maze test of spatial memory. Also tested this in associative memory in the fear conditioning tests. And the same here, we could see that both males and females perform better in associative memory tests. Here we have the contextual test and followed by the Q test in the following day. One thing that is maybe interesting here is that there's no real effect of menopause or surgical menopause or ovarectomy here. Actually, there might be an inverse, like they might be better off with ovarectomy than not having it. Then we'll also look at A beta plaque levels, and we see that actually there, especially in the hippocampus, that we get less plaques, but also smaller plaques, both in males and in females. And also ovarectomy had some effect here, but not had no adverse effect here. But we saw that LOL ligand had an effect in lowering plaque levels, both in the sham and the overactomized mice. Something interesting also that we see always generally higher A beta levels in females than in males. Here you show the cortex soluble and hippocampus soluble A beta 42 levels that are always higher, you see, in age-matched females compared to males. So then we want to understand what is the cause of this of lower amount of plaques. And we checked APP processing. So as you know, APP can be processed in the amyloidogenic pathway with beta secretase and gamma secretase to give rise to, at the end, A beta that can aggregate, or it can be the non-amyloidogenic pathway with alpha secretase and gamma secretase. And what we checked here was beta CTF levels and also the full-length APP levels with Western blot. And then maybe you can just focus on this middle part where we look at the beta CTF levels. And as you can see, there is no big effect or no change in beta CTF levels in any treatment or with ovrectomy. We don't see anything. One thing that might be interesting is that we have, in general, higher full-length APP levels upon ovrectomy, which we don't really know if we have that, why we have that. But there's no effect on processing overall. Then we ask, okay, if there's no effect on processing, why are there less A-beta levels in the mice upon A-beta activation? So could A-beta modulate neuroinflammation, A-beta scavenging instead? And as you know, microglia is a very important A-beta scavenger, and it can be activated with amyloid beta oligomers, and it can this is very good for removing phagocytosing A-beta oligomers. But when the disease progresses, you have the microglia simply cannot cope with this, and they get overactive, secreting pro-inflammatory cytokines, and this is then damages the neurons. So we look at neuroinflammation, and just I show that two here, pro-inflammatory markers that were downregulated in the males upon A-beta activation with this LY ligand. And these two, CXCL1 and IL-12, some classical pro-inflammatory markers. But in the females, we don't see the same effect with CXCL1. With IL-12, we see it with LY treatment. But ovrectomy seems to sustain high neuroinflammation levels in the females. We also see the same with IL-10 here, and IL-10 is a sort of a death signal that tries to counteract pro-inflammatory markers, so it's coming in quite late. So generally, the males have less neuroinflammation, we can say here, and ovrectomy sustains high neuroinflammation in females. So then we looked more specifically on microglia, and we could see that we have less overactive microglia in different brain regions. We have looked at frontal cortex, the somatosensory visual cortex, but also in hippocampus. And in hippocampus, we see the biggest effect with less activated microglia in the males, which coincides actually with more plaque-associated microglia. So there are less microglia that are superactivated, and they are more functional upon LY treatment. In the females, we see similar things, but to not the same extent regarding activated microglia, but we see no effects on plaque-associated microglia with A-beta activation. So we see stronger effects in males than in females when we activate A-beta on microglia. Then we look also in A-beta how much expressed it is in microglia, and it's actually only 5 to 10% of all microglia that are A-beta positive. But those that are more positive, they are in the males compared to females. So we think if only 5 to 10% of all microglia are positive, then other factors or cell types are also involved. And to study this, we decided to knock out A-beta in this AD mouse model, the APP and LGF mice. And here we started this project some time ago and still ongoing. I will show you the data on the 6-month-old APP knockout mice. A-beta knockout mice in the APP model. We use both males and females. And actually, we don't see any effects on memory when we knock out A-beta. None on YMS or other memory tests. But we see an effect on A-beta plaque load or the plaque area, where in the males that lack A-beta, they have higher amyloid plaque load than females. Than wild-type mice. In the females, we don't see any big effects, although it's maybe a tendency to higher plaque levels in the knockout. Then we decided to perform single-cell RNA sequencing of these mice to understand where A-beta functions and what it does with different cell types, why we have more A-beta plaques in the males and not in the females, for example. And we can see that if we just compare male to female, wild type, but these are NLGF mice, and male to female knockout NLGF mice. Actually, we don't see big effects at all. Most of these genes are X or Y chromosomal genes that are differently regulated. But then we also look at ER-beta knockout effect of ER-beta knockout here. And we see that actually here we see now very many genes that are differently regulated, both in females and in males. So knockout of this single gene ER-beta, you have massive effect on gene expression in both males and females. So then we went back a bit to look at ER-beta in the brain and what it does there. And we know that from early life that ER-beta is very important for radioglia migration, cortical layering. It can regulate BDNF signaling and also neurogenesis. And neurogenesis is also something that is ongoing quite more in mice than in humans, especially in the hippocampus. In the adult mouse, we also know that ER-beta can regulate synaptic plasticity and also oligodendrocyte resilience. But in older age, we don't really know so much about ER-beta function. We know that there have been some studies that have shown that it can modulate AD pathology, and it has been shown to maybe participate in neuroprotection and mitochondrial function. So then we look at which pathways are affected when we knock out ER-beta in this AD mouse model. And probably you cannot see all of these small text here, so I kind of just highlighted it. And if you just look at all genes, all clusters, both males and females, just ER-beta versus knockout. ER-beta knockout versus wild type. We see that downregulated genes belong to actin cytoskeleton organization, and upregulated are those associated with synapse function, exocytosis. And this is also something that we have seen in male mice, at least, that there is an imbalance in excitatory versus inhibitory transmission upon ER-beta knockout. And we see also that there are more vesicles in the excitatory neurons than in the inhibitory neurons. So there is sort of imbalance here. And we also see that the mice are all more, they have sort of a hyperactivity, and also they have more spontaneous electrical activity in the brain. So then we stratify this looking at males, and again here maybe you don't see. In males, downregulated, both out but downregulated were genes associated with DNA repair and cell stress, while in females, the genes upregulated were all associated with mitochondrial process, and only one gene that was downregulated, or one pathway that's downregulated was associated with insulin secretion. So it appears that also glucose metabolism and mitochondrial bioenergetics are important here, but also maybe cell stress. Then we decided to look more specifically on the different cell types, which cell type specific pathways affected, what are differences between male and female in these different cell types, what's the different effect of knockout versus wild type also. So we started with looking at microglia population. Again, we don't see a big effect on sex, female versus male here, but we see an effect on knockout of ER-beta again here. Looking at the cluster here for microglia population, we can divide it in homeostatic microglia, disease-associated microglia, and the more advanced disease-associated microglia. And what is interesting here maybe is that we have one population that is different in knockout compared to wild type microglia, both in males and in females, while the disease-associated microglia were mostly the same, actually. And looking closer with this microglia population, we could see that apoptosis, cytoplasmic translation, and protein folding and stress response was affected pathways upon ER-beta knockout. So it appears that ER-beta knockout can knockout of ER beta can result in more vulnerable microglia. Then other pathway or other cell type that was very interesting to us was the oligodendrocyte population. Again, we didn't see a big effect on just looking at female versus males, but again, we see a bigger effect of knocking out ER beta, both in females and in males. Looking at the pathways here, looking at the different oligodendrocyte populations here, we have the OPCs, the oligodendrocyte precursor cells. We have also early myelinating cells and late myelinating and mature oligodendrocytes. And there was no big effect, no differences in mature oligodendrocytes, but we saw differences in the numbers of OPCs, and also early and a bit later myelinating oligodendrocytes. Looking at the pathways here, we could see that in females, basically was not so much affected, only locomotor activity. And the males upregulated was myelination and insulin signaling, which we think is related to myelin synthesis, which requires a lot of insulin. And then we revisit all the ER beta knockout data, and we could see actually that this kind of makes sense that ER beta males are, at least males here, we only have male data from this old study, that these males are more motor active and also display hypermyelination, and they have thicker but a bit more defective myelin sheets with these blebs in the sheet. And then we also looked more on the genes that are expressed here, differently expressed, and actually one gene stands out quite a lot here. It is a gene called TTR, which is actually totally differently regulated between males and females and between knockout and wild type. So it's upregulated in female knockout and male wild type, and downregulated in male knockout and female wild type in the GF mice. So what is this TTR? It's called transthyretin. It's a transporter for thyroid hormone T4 or thyroxine in the blood, and so it transports TTR around the tissue it transports thyroxine around the blood from the liver, and it can transport it to the brain also. And in the brain, it can actually bind to amyloid beta, the amyloid beta peptide. And in this way, it can inhibit A beta oligomerization and decrease A beta production, and also affect A beta cleavage. So it's a protective factor in the brain. And it's highly expressed in choroid plexus and also in the oligodendrocytes. And 80 patients have shown that there are lower TTR levels in the CSF and serum. And we see now in this A beta knockout male APP and LGF mice that they have lower TTR expression, more A beta plaque levels. So there is also another publication that showed that loss of TTR results in hypermyelination and increased differentiation of neuron stem cells to OPCs and reduced GABAergic signaling. And they showed also that when you have this knockout, that you have more oligodendrocytes in males, but not in females. So this is very similar to what we see actually, that loss of ER beta in males, you have less TTR expression, you have hypermyelination, you have increased excitatory transmission. This could result in excitotoxicity and maybe also neural vulnerability. It's something that we want to study more in detail now. So to summarize this, I told you that A beta can protect against AD pathology in the APP and LGF model. It warrants also more studies on A beta selective agonists to combat AD, but are cell-specific sex differences. And it seems that male benefits actually more from A beta activation than females. So maybe we have to also rethink if A beta agonist should be given in menopausal hormone treatment. Maybe not, because it doesn't affect the female as much as males, but still we have some effect in the females. The mechanisms are also largely unknown, But it could be like affecting factors like this TTR in a cell and sex-specific manner. And also one thing to remember is that mice are not human, and we need very much translational studies to confirm the role of ER beta activation in humans and in human pathology. Yes. And with that, I want to thank you for listening, and I want to also acknowledge Aphrodite, Heba, and Michael in my group that did most of these experiments. And this is my former group members, and these are my collaborators and my funding sources. So thank you for listening.
Thank you, Ivan. That was a great talk, simplified, so everybody can follow, which is good because the audience of the HIAAD seminars is really broad, as I told you. That's very good and really great work and very nice presentation. I'm sure Yanis Haralabopoulos have at least 16 different questions. I have like 20 also. And Savas is looking at me that he wants to say hundreds of things, especially because they're also focused on amyloid beta, part of AD pathology, and different parameters like inflammation that they work in their lab. We are more in tau, Christina in the depression. So Yanis, I see also Kostas Aliketsos is here, a lot of people. So let's start with Yanis because I know he's willing to.
Yes. Actually, you know, Ivan, even if we co-edit these frontiers, the main work in our loop is of my group is not with glucocorticoids, it's with steroid derivatives from DHA actually, and a little bit of estrogen in Alzheimer's disease. So your talk was absolutely interesting, very interesting. And one quick question, since you ended your slides with this, have you checked, you or others perhaps, the expression of ER beta in iPS-derived neurons from AD patients compared to control and so on?
No, not in AD patients. No, we have not. And I don't think anyone has done it in patients. Yeah. No.
Okay. Very interesting.
I have a collaborator that is working on this, but he has not checked beta levels yet.
Okay. Do we know if, I suppose they express ER alpha?
Yeah, I mean, IPSs, they express not so much ER beta or ER alpha, but once you differentiate those, they start to express a lot. It doesn't matter to what lineage ER beta is highly expressed. And especially maybe if you differentiate to neuronal lineages, ER beta goes very high. ER alpha, not so much, but ER beta goes very high, but then it goes down. It goes down when you have mature cells, it goes very much down. So it's only low expressed, and not all cells express it either. Not all neurons express it.
Really interesting. And the other thing that I was thinking, presenting your data with the mouse, transgenic mice, have you checked also the adult hippocampal neurogenesis after the administration of the agonist?
No, not yet. No. This is something also we want to look at.
Because ER beta is also expressed in the
it's
the adult neurons themselves.
Yeah, and we are looking at the dendrite gyrus. We are trying to actually look at PAX6 staining in the dendrite gyrus, which is like the neurogenic cells. And right now we don't see big effects, big difference between treated with LOI ligand cells. There's no difference there. But we have still to look in these knockout mice if we have differences in PAX6 staining in the dendrite gyrus, for example.
Or, you know, during the differentiation, even after the PAX6 and SOX2, to check for double cortin and things like that, to see if the process during maturation is also changing. And one last question, not to bother you too much. Did you try to change the time of administration, I mean, earlier or later in this animal model in order to see if your effect is preventive or it has something to do, you know, with the progress of the disease?
No, we chose, we started to treat the mice around 3 months of age, something like that. And this was decided to put this because then when we then analyze the mice at 6 months, we will hit the curve when it's like on the exponential growth of the pathology curve. And then if we see effects there, we will see the biggest effect. So this is why we chose like more or less this time point, 6 months, to look at the pathology. But we have also another model which have slower pathology, and there we want to treat it maybe for shorter maybe time points periods, but more like over longer time. But we haven't done that yet. It also depends a bit on the, on the model and everything. And this model is like 6 months of age. They are still not aged, I would say, but they have pathology like a 90-year-old familial lady.
Yes.
So it's only a pre-model, I would say.
Since you are seeing differences in the cognition, that's fine.
Yeah.
Okay, thank you very much, Ivan.
Is next Sava?
Yes, if I can ask. Thank you.
Yes, please. Yes, please.
So I was wondering, I have a general question and a more specific one. So I was wondering whether patients, women that suffer from breast cancer and then they get aromatase and inhibitors as a treatment therapy against estrogen receptors, against production of estrogen, are they more in danger of Alzheimer's, these patients?
From looking at the sort of evidence in other areas of this, I would say yes, but there has been no study directly addressing this point. So this is not well known, but from looking at the other literature around this, I would say there would be, yes. Yeah.
And
the only
similarly,
thing is that they maybe don't survive so long anyway. If yeah.
And similarly, since statins are so popular nowadays, I mean, people that get statins and reduce their cholesterol levels, which is the substrate for producing then estrogens, are they also more in danger? Are these concerns looked at? Are they studied?
I think it's not been studied so much. I don't know anything about this actually. But I would say this is there's a risk versus benefit here also.
Right.
And Alzheimer's disease is a very late onset, well, not always, but often a late onset disease when you're aged. And if you have like a prognosis that you live maybe 10 years or 20 years, then you take the whatever medicine you get to alleviate first, like cancer or whatever. Yeah.
And the last question, the data about the TTR are very interesting and very impressive. So this protective effect of TTR against A beta and the transcytosis of A beta through blood-brain barrier, have you looked at it, how this happens? What is the mechanism? Is this through endocytosis in endothelial cells and then secretion in the bloodstream, or how does it work? Is it passive diffusion through the junctions of endothelial cells?
No, I think it's the active transport through the blood-brain barrier of this transporter, basically, that carries then the hormone.
Okay.
And in the brain, it has also affinity for A beta, and there it's kind of sort of takes the beta away from getting aggregated. This is how.
Yeah, that's what I'm asking. Basically, how does this work? It's through the pores?
This is not very well known actually how it works, but the transport over is active, I would say.
And also these fenestrations in endothelial cells, perhaps they are also involved. Yeah,
it could also be, it also has this, it says here, vascular protection, so it's also like that probably associates very much to the endothelial cells here. And it can also be like it's not been studied very much, the TTR in relation to Alzheimer's disease. I think it's like 3 or 4 publications only. This is one of those, and it could be also that it's protecting against vascular dementia more than maybe other dementias or like A beta aggregation around the vasculature also. Yeah.
Very interesting. Thank you very much.
Do we have more questions? just very fast, are you thinking to check this with tau or any downstream, because, amyloid beta is one key component, and we know that it is essential to start. But don't you think that it could have similar effects on tau?
Yeah, we don't know yet because this mouse model only has amyloid pathology.
I know.
But we are now developing, or collaborators develop one with tau pathology, also with tau, or has two mice models now with just overexpressed in the human tau gene and one with the mutated tau gene. So we're also very interested to check a beta treatment or activation treatment of these mice. And we also have some organoids that are...
Are you doing that with Per Nilsson?
Yeah, Per Nilsson, yeah, exactly.
Very well.
He's the APP and NLGF guy here in Sweden. So, but we also have organoids with NLGF mutations, human, and they were there. We see actually there is effect on tau.
Per is a nice colleague. We were together in Riken many years ago, and he's more interested now in tau. Yes.
Yeah, exactly.
He contacted me already. Okay, great. So you are doing it. We will know soon.
Yeah.
Okay.
Takes some years still, but soon.
Thank you. Any more questions from the audience, or Christina wants to? Christina?
Yes, hello. I don't really have a question. I just want to say thank you very much and congratulate you for this amazing work, because now I saw many things that you didn't present last time. So I see that you have many different
it progresses.
lines of research and projects. So it's very exciting. And I hope we can all collaborate at some point in the future.
That would be great.
The Greek colleagues. And also, I remember last time that we talked about a new project that I'm sure it's too early maybe to talk about it, but it was very exciting about the gender dysphoria in humans, of course. And it might have a link with Alzheimer's disease in some extent. But,
maybe I can explain a bit about that project. It's different from this, but we also look at sex hormone treatment with sex hormone early in life, like at when you're like in 20s, and we look at the gender dysphoria population when they are treated with gender affirming hormone treatment. So males that receive testosterone blockers and estrogen supplement, and females that receive testosterone. We know there will be probably effect on the cardiovascular disease from that. I think no one has checked that, but also maybe on AD risk later in life. So this is now also very hard to study because we have no cohorts that are aged from these hormone treatment groups. This is something that we're looking now into with using epigenetic approaches. So maybe in 1 year I can update you on that, maybe with some results even.
Yes, we hope so. It sounds very interesting. Thank you very much. Thank you. And I hope next time you will be here in person.
Yeah, that would be great.
Yes.
I don't know.
Ivan, may I ask something irrelevant?
Yeah.
Ivan, are you were in Zanaki's lab with Christoforos Thomas together?
Yeah,
Working
yeah.
Christoforos is a good friend. We met recently.
Okay.
He's in the States now.
Yeah, he's in the States. Yeah, I did my PhD, and he did his postdoc in the same lab, but it was 2006 or something like that. Yeah.
He works in Nalvarte.
Yeah, still in Nalvarte. Yeah.
We are very good friends when we are both in Karolinska, so I know him well.
Yeah, he's a nice guy, very funny guy.
Okay. Thank you.
He comes from Ioanna, from here.
Yes,
right. Right.
Makes a lot of advertisement for you, Ioanna.
Great. I'm happy to hear this.
You can stop sharing so we can close this today's seminar. If there's no more question, I would like to thank everybody for being with us in this first seminar of this new academic year for the HIAAD, and hope you will be with us next month as usually. The first Thursday of the month. Today was an exception because there was summer break for many of us. So thanks again, Ivan, for joining us, and see you next month. Goodbye.
Bye.
Bye. Thank you.
Bye.